We are going live, ma'am.
Okay.
Very soon. Okay, now we can start, ma'am.
So,
We are going
good afternoon to all of you. So we are going for starting the phase first program session. So it's my to all of you. It's my great pleasure that I am introducing to Dr. Vandana B. Patriwala is currently a professor of pharmaceutics at the Institute of Chemical Technology, Mumbai, India. She has around 30 years of the teaching and research experience. She has over 200 referred publications, H-index 43, I-10 index 103, 14 granted patents, and 27 patents in pipeline and 3 trademark registry and 1 industrial design. She has published 2 books and 32 book chapters with international publisher. Dr. Patriwala has been active in teaching, research and service throughout her career. Her area of research include development of nanocarrier with a major emphasis on the infectious disease, cancer and neurodegenerative disorder, medical device development, nano diagnostic and nano vaccines. She was awarded to Kreza Oriation Award 2020 APTI and Dr. Manjushri Pal Best Pharmaceutical Scientist Award 2009, Shri Amrutamode Distinguished Research Award 2018, OPPI Women Scientist Award 2015, Bill Melinda Gates Grant Award 2015, Best Pharmaceutical Scientist Award 2014, Vasvik Award 2013, and Benito Nanotech Award 2013, APTI Best Teacher Award 2012, Fellowship of the Maharashtra Academy of Science 2011, and KH Garda Distinguished Research Award 2009. She is a Vice President of CRS Indian Chapter, Editorial Board Member, DDTR Editorial CRS, IC, and APTI Women Newsletter. She is actively collaborating with the researcher as well as industry within the country and abroad, and has completed Indo-Swiss, Indo-Japan, Indo-UK project. She is executing all the major grants from the Indian government focusing on the product development. She has transferred about the 20 technology to industry, including drug-eluting stunts being marketed in a more than 16 countries. So there is such a big resumes are of work of the this our reputed ma'am. So, but I will just do the brief here. So I would like to invite to deliver your way, ma'am.
Thank you, sir, for those kind words. And in fact, at the outset, I would like to thank Shobit University and yourself and Dr. Niladri Ghosh for extending me an invitation to, in fact, deliver a talk. And if you could confirm, my slide is visible.
Yes, it's really visible, ma'am.
Okay, so my talk today is on ICT tools in pharmaceutical research and development. And in fact, today I would be dealing with some of the case studies which we have done at ICT. What a coincidence. A person from ICT, that is Institute of Chemical Technology, delivering lecture on ICT tools. So in fact, pun intended, but definitely here you can see our glorious institute, Institute of Chemical Technology. For those of you who are not conversant, please, I welcome you all to visit Institute of Chemical Technology, which was established in the year 1933. In fact, this is one institute which has given 19 Padma awardees, which is highest civilian award, as you know, of our country to the nation. So once you enter these portals of excellence, the Vastu is such that you start working and giving your best. So currently, yes, I am in Department of Pharmaceutical Sciences and Technology and working in capacity of Professor of Pharmaceutics. So what are we going to do today is I'll introduce the concept and look at what are the computational tools which could be used in pharma as a whole, be it in research or be it for industry, introduce some of the computational pharmaceutics as well as molecular dynamics simulations. You will see that how on basis of in silico work which we do at ICT, we have designed various nanocarrier based drug delivery system. And essentially our group, which we term as VBP research group or VBP family, goes after my initials. In fact, we are working a lot in the area of polymeric and lipidic nanocarrier systems. So you will see few of the case studies from my own group, besides the studies which have been done in literature. And of course, finally, we will conclude. So this is a roadmap of my talk. Looking at this slide, it gives you a very good idea as to what can human brain do and what human brain cannot do. It's actually, it's limitless. You can do everything provided all areas on your frontal and the parietal lobe are clear. So basically, we know that nowadays research is becoming more and more multidisciplinary. So people who are in the pharmaceutics, obviously, if you look at only pharma, they'll be collaborating with pharmacognosy people, pharmacology people, also pharmaceutical chemistry people, but it is not limited now. You can collaborate with biophysics, biochemistry, and colloidal sciences. It could be anything. Basically, what we are trying to show here is that it is a modern era of interdisciplinary research, and more so in this time when COVID has hit us so hard. Our students, believe me, sitting at home are actually doing work on in silico models and coming out with strategies to treat COVID-19. So they are looking at various databases. Now in Government of India has announced hackathon, so people can work in their own houses and work in way so that by the time institutes reopen, they are ready to synthesize those molecules or modify those molecules and test it. So this slide just indicates that we can do interdisciplinary research, and when we deal with nanocarriers, which is our specialization, colloid sciences is one field where understanding of biophysical properties really comes to our savior. So what is the role of these tools in pharma? If you look at what it was in the past, we were looking at descriptive models, which gave us some leads. But we are moving now towards predictive models. And that is possible only because we are able to make use of artificial intelligence. So whatever data is available, be it in literature or be it whatever you create, you can actually model so that you can prescribe. So from descriptive, we are moving to prescriptive future. And as you can see that value of this research is going to increase when we move towards predictive models, and obviously when the complexity will also be more. Obviously, you need to have workstation which can work faster and faster. We cannot, in fact, make the importance less of ICT tools. In fact, if you look at these Nobel laureates, Martin Karplus, Michael Levitt, and Dr. Warshall, in fact, they got Nobel Prize in chemistry in year 2013. And what was that for? That was for looking at how protein-protein interaction take place. And as you know, proteins are large moieties. So it was virtually impossible to do experiments and look at how all atoms will interact with other atoms. So what is their statement? They said, today, the computer is just as important a tool for chemists as a test tube. It conveys a lot, says that experiments are important, but nevertheless, you cannot deny importance of computers, and that's where ICT tools come into picture. So you will see in the lecture which follows, we'll be focusing on computer-aided drug design, but my area majorly, because we are working in drug delivery systems, which is a formulation science, deals with pharmaceutics, and therefore it would be on computational pharmaceutics. What are various computational tools which are used in pharma? If I have to list them, this is how I would list them. So you can classify them on basis of ligand interaction and molecular dynamics, where tools like Schrodinger, BioSuite, Autodoc, Gold, etc. can come. Or it could be where you are studying, structural activity relationship and making use of molecular modeling, wherein you can have Maestro, Gram, PAS, etc. Nowadays, even pharmacokinetics could be predicted. So if you can fill in data in silico of release kinetics, it can predict whether your system will get absorbed or not on basis of physicochemical properties of that particular API and the delivery system which you have selected for them. So the tools which are used include GastroPlus, DDD Plus, and MapCheck. In fact, pharmaceutical industry nowadays is using this GastroPlus for pharmacokinetic parameters. And on this side, people are using Schrodinger, which is providing a very good, user-friendly model. And of course, if you have to do data analysis, be it clinical data or be it proteomics or anything, you can use something like GeneSpring. And then there are other ones like QSAR Pro, EthoWatcher, etc. And if you are working in the area where, in fact, you are doing, say, drug delivery to brain for, say, Alzheimer, Parkinsonism, etc., you need to track what happens to those animals, first in animals, in preclinical studies. So those can easily be tracked using EthoWatcher. So people have actually designed, in fact, they have written these software programs. They've written algorithms, in fact, to run these programs. And without going into intricacies of that, for time being, it's important for you to understand that you have plethora of tools available. And depending on what you will be working, you can select any of these, and most commonly used ones are listed here. So Schrodinger, Material Studio, and GROMACS, people are using a lot. And in fact, it's a molecular dynamics when we say it is how atoms react with other atoms. So there has to be a trajectory which needs to be followed for those atoms to interact with the atoms. And that definitely depends on Newton's law of motion. So what is the force? What is the interaction energy? All that is actually computed by molecular dynamics. So such a thing, if the tools can do, obviously you can get lot of information from this data. So what are the categories of simulation models? And what are the spatio-temporal domains of applicability are given or depicted in this slide? Of course, the question of interest will dictate the level of resolution which is required. And as you can see here, if I were to plot time scale on y-axis and length scale on x-axis, the level of detail decreases in this direction. So you do have quantum mechanism, you do have AA model, which is nothing but basically atomistic. Then you have CG, which is coarse grained, and you have DPT, which is dynamic model, which needs to be looked at. But you can see that level of detail increases in this direction. That means this quantum mechanics, which will actually be looking at how electronic motion happens. Not only atom to atom, but electronic motion can be tracked. So that is what you get, information you get using quantum mechanics. Obviously, because level of detail is more, it is highest, in fact, very, very high cost one. The one next is AA, which is nothing but atomistic or all atom, wherein you are looking at how point each atom reacts with another atom. So they remove some of the important things like what bond formation is taking place, what are the cleavages, etc. All that is removed as compared to quantum mechanics. So obviously this is much more preferred. Then comes AA, and then there is CG, which is coarse grained. And in fact, most of the pharmaceutics related work, even coarse grained and atomic, all atom is okay. In coarse grained one, what you're doing is you're treating group of atoms as one interaction point. So all hydrophobic ones will be grouped together. And then you can actually look at what are the electrostatic interactions or what are the hydrophobic interactions. And in the DPD model, basically, you are looking at both the dynamic and fluid flow properties. So from atomic, you're moving to the molecule level. So this is molecular level. Here all are grouped. Here atom reacting with atom, and here electronic transitions which are looking at. So you can see how then the time scale decreases. With quantum mechanics, what you can do in picoseconds, with DPD, you will require milliseconds. So obviously, you require specialized computers with high capability to do this job. So just for those of you who are not conversant with what is molecular dynamics and what kind of atomistic MD simulations are there, I would like to take you stepwise. So we can divide them into three. One is the pre-processing stage. Next is the molecular dynamic stage, and then you are post-processing it. So first step actually is to construct a molecule. All this, believe me, is being done in computer, so you do not need any wet lab experiments. So what are you doing? You are looking at initial geometry. So either you can take up this molecule if it is available in protein database, you can directly import it from there, or you can actually draw it using ChemDraw. So first you are constructing your molecule because you are aware as to what you are synthesizing or what you are making or what is your API. So that easily can be constructed. Then you are looking at definitions of interatomic bonded and non-bonded force. That means once you have molecule, you will have atoms. Once you have atoms, you know what force field would get generated, how that atom will react with the other atom and with the rest of the atoms. And that is the concept of your force field. Then actually, depending on shape and size, depending on boundary conditions and filling molecules, you are creating a setup. So it's like a setup box. So once you have fed in the software, in fact, if that is available to you, all you have to do is create a box wherein you will be putting your molecules which you want to use in research. So if you are wanting to look at how my nanocarrier is going to react with phospholipid, you will be putting your API, which is in the nanocarrier. So whatever material you have used, lipid, and then you will also be putting your phospholipid. So all that would be put virtually in a box. And then in case, there is a provision given even to put filling molecules. Like for example, sometimes, for example, antibacterial activity, etc., zinc plays a very important role. So if you want to put some such Ions, you can put them too, cations or anions, etc. So you are putting your molecules, and in addition, you can put even these cations or anions, which may be playing a very, very important role as per your hypothesis. After that, the next step is to remove the overlap between molecules. Maybe your molecule is a salt form, maybe it is a hydrate form where water molecules are not playing a role. So you will be removing all those molecules which are not very important, and therefore you will be actually minimizing the energy. And once energy is minimized, obviously you are allowing this setup box to play for some time. In fact, if you want to publish in very high impact factor journal, they say that you need to run what I have shown here on the right hand side is how three-dimensional molecular dynamic simulation is done. So this should run for 500 nanoseconds. But just for screening purposes, we actually virtually run it even for 20 nanoseconds. In 20 nanoseconds, you can get a very fair idea as to which particular molecules I should go ahead with and which ones I can delete. And this will go on simulating till it reaches thermodynamic equilibrium. Just to give you an idea, when I say 500 nanosecond, in fact, it doesn't take that only 500 nanosecond, it takes 3.5 months to tell you, give you the information which will be at the quantum level. So it does take time, whereas the 20 nanosecond one is just 2.5 days job. And then once you have that thermodynamic equilibrium, MD studies have been done. That means it will record coordinates, it will record momenta of atoms, of course, at predefined intervals. So these you have to set. The intervals you need to set in your computer. And then you will be analyzing it. You will be determining whatever properties are of your interest from the simulation trajectory. So I think if this is clear, these are the basic steps for all atomistic molecular dynamic simulations. From software to software, some of the terminologies which are used will change, but basic steps obviously remain the same, starting with construction, looking at force field, looking at the box setup, doing energy minimization, equilibrating it, and then processing it, finally analyzing it. Now coming to application of this in drug delivery systems. Where can I use such a data? First is you can do physico-chemical characterization. So one can actually look at how a drug reacts with whatever drug delivery system is in question. One can look at what is the drug loading and distribution, all in silico. Look at the aggregations which may take place, leakages which could take place. So it gives you idea about stability, be it for suspensions, be it for protein aggregation, anything. Besides that, it can give you idea about whether your system will self-assemble. So whether it can form micelles, whether it can form dendrimers, and give you idea about structure as well as dynamics of the complex. Not only that, one can look at its interaction with all the membranes. So it could be, as I said, it could be liposomal membrane. You want to see whether liposome is the right kind of moiety for my drug delivery system when I'm making, or should I go with neosomes or ethosomes? So if you have to do that, even that can be done in silico. So you can also look at whether internalization can happen cellularly. So when we talk about endocytosis as a pathway, whether it happens virtually also it can be seen before whether it can be tracked in vivo. As far as circulation is concerned, one can look at all the interactions with ions. I just gave you example of adding zinc. That can be done. Same way you can add any other anions or cations. So that will give you idea about the stability, and it will give idea about also interaction with different biomolecules. At the intracellular level, though, one could look at whether once it goes into the cytoplasm, it gets localized in the cytoplasm, or it passes on and reaches nucleus. So even that kind of information is possible by looking at, typically in pharma, what we look at is van der Waals forces, we look at electrostatic attractions or repulsions, and only these things itself can give you so much information about what would actually happen in vivo. So I'll take you through each of these now. So computing drug physicochemical properties, you can see that one can do solubility calculations. So we all are aware with Henderson-Hasselbalch equation as well as Henson-Hildebrand solubility parameters. And this in turn is based on Florey-Huggins theory. So we can determine solubility in silico. We can look at miscibility of ingredients in silico without doing experiments, and then also look at its thermodynamic stability. So glass transition temperature easily can be recorded in silico. And then you can look at how chemical degradation takes place and also determine partition coefficient. So all these plethora of activities without doing any experimentation. And I'm sure all of you are aware that these properties, in fact, govern a lot as to which particular nanocarrier I'm going to make. First thing which we do is what is the solubility of my API? Next step I look at is what is the partitioning? So if solubility is less, that means partition coefficient is very, very high. I know that this moiety either belongs to BCS class 2 or belongs to BCS class 4. So knowing that, obviously looking at the structure, we can always say whether it will undergo chemical degradation in presence of acid or base or oxidation, what kind of degradation mechanism it follows. And then if we know its thermodynamic stability and also know its melting point, both these properties can help decide whether I will go for lipid system. If melting point is less than 140, obviously I'll go for lipid-based system. But if melting point is very high, say it's about 200, 230 degrees Celsius, we know that that mode will not work. So in silico determination itself is giving you idea about what type of dosage form I should be making. So this is one example where miscibility of indomethacin was studied in different carriers, and then in silico correlation was done using thermoanalytical technique. So what was used for molecular dynamics? Compass force field was used. Cohesive energy density, which refers to energy required to vaporize a mole of liquid per unit volume, was determined, and solubility parameter was calculated. Solubility parameter is nothing but square root of this cohesive energy density. So if you look at this data, this is for indomethacin, for polyethylene oxide, glucose, and sucrose. Now look at MD simulations, that's molecular dynamics data. Very clearly, it indicates if the value of your excipient is close to that of API, that means it will be miscible in that excipient. If the value is far away from the API value, it would be immiscible. So if it is very close, as you can see in this case, indomethacin value is very close to PEO and therefore it is miscible. Sucrose lies somewhere in between glucose and PEO. So having done this, a very simple experiment in silico has given you idea that if you want to solubilize your drug, you can go in with PEO. So you can select excipients and go ahead. So likewise, you can screen 10 to 15 excipients and look at this data and select those excipients where these values are very close. And if you look at this particular data, here the difference is 10.9. 10.9 itself means that it is far, far away from indomethacin, closest being PEO, sucrose being in between. And then once you have done that, in fact, in this particular paper, once they did that, they have compared it with thermoanalytical technique, which was differential scanning calorimetry. So obviously, now that if we put indomethacin in PEO and it is soluble, if you record DSC of indomethacin, you will get peak of indomethacin at its melting point. But if you make indomethacin PEO combination, since indomethacin is soluble in PEO, you will not see any peak of indomethacin. So likewise, you need to interpret your data. This also could be used to all kind of lipidic drug delivery systems, polymeric delivery systems, dendrimers. Nowadays, a lot of work is being done on co-crystals for solubilization as well as for retardation. One can look at protein and peptides delivery systems also, their aggregation properties, as also one could look at inorganic nanoparticles too. So meaning sky is also not the limit once you talk about using, computational simulation. It's only that your hypothesis has to be very strong. You should know what actually are you wanting to do. Once you know that, definitely you will reach your goal. So generally, how lipid-based drug delivery systems are represented is depicted in this slide. So earlier we talked about atomistic model, that's all atom model. So here it is actually schematically depicted. So you can see that this is a fatty acid chain. So this is fully atomistic. When we talk about united atom, that means grouping has been done. You can see that now this structure is simplified. From here to here, it is simplified. And now when you go to coarse-grained, it is further simplified because you are grouping the similar ones together. So if you go on from here to here there are 149 interaction sites. And in coarse grain, there are only 12 interaction sites. That means your work gets simpler when you move from fully atomistic to coarse grain, but you should understand your molecule very well. Then only there is, in fact, importances of going to coarse grain. If you do not understand everything is new, it's better you work with fully atomistic system. See now, liposome, all of you are aware, liposome, which is used as a drug delivery system, is a bilayer model. And what is that bilayer? Obviously, it has a hydrophilic head and it has in between a non-polar chain, which is hydrophobic. And then depending on your moiety, whether it is hydrophilic or it is hydrophobic, either it would be put in the hydrophobic domain or it will be in the hydrophilic domain. Depends on the solubility of your moiety. So they have simplified this full structure by just depicting it as hydrophilic head, hydrophobic tail, and then again hydrophilic head. So this is your coarse-grained model, wherein you are simplifying things rather than looking at very, very specific chemical details. So from this, the measurable properties which you can get are with respect to permeation. You can get what kind of dynamics is featuring into when interaction is taking place. And obviously, you can get idea about structure also. I'm sure all of you who are working in all kind of nano delivery system would understand the importance of packing ratio and how this packing actually influences drug encapsulation and leakage. We do encapsulation efficiency to determine that. But can it be done in silico? Answer is yes. So you can measure using in silico all these properties. And this slide very, very clearly depicts the model membranes and what job they can do. And you can see that in this particular slide. What I have shown is how stratum corneum is going to interact with lipidic membrane. So not only that, you can look at how liposomes are formed, how your drug would interact with these liposomes, how your skin reacts with these lipidic membranes, whether the molecule which is in question, which is actually put into liposomes, when applied to skin, whether it will be taken up by skin. So you can look at even the uptake. Not only that, whether interaction is taking place between any kind of nanoparticle, be it polymeric or inorganic, and the phospholipid membrane or your skin membrane. All the information you can get in silico. In fact, people have also elucidated what is mechanism of action of penetration enhancers. We all know that penetration enhancers, typically the ones which are used, like say ozone or dimethyl sulfoxide, dimethyl formamide, etc., we say that it fluidizes lipid membrane. So for doing that, will you be applying it on skin of humans and then looking at actually porosity increases or not? Answer is no. Easier thing would be to do it in silico and see whether the pore opening is taking place or not. And that easily can be done using computer. So sitting in your four walls, at home, you can do a lot. And in fact, you can publish it in very, very high impact factor journal. You will be surprised if I tell you that during this COVID period, 3 of our papers have got published from the lab by undergraduate students. So undergraduate students are taught computer-aided drug design. So they have actually looked at a lot of herbal moieties, and from protein database, structure of receptor for COVID is available. So they have looked at which moiety will interact in the best possible way with that receptor and proposed a drug delivery system for that. So all in matter of 6 months, 3 papers from undergraduates. Of course, there are 9 others from our research group, but I thought it would be good to indicate that even undergraduate students, if they are given access to such softwares, and they, believe me, some of these softwares are free. Schrodinger is paid, of course, but some of them you can, in fact, the model, which is not the latest one, you can actually download it for free. So, and then work on it. So that's how our students are working, because they are not able to go to institute and work on it. Now, this is one example wherein it is elucidated how endocytosis of nanoparticle happens and how pH modulates that. So you can look at these. When pH is low, the figure A is a figure when pH is low. That means ionization of this particular polymer is low. As a result, the polymer which you are actually using does not very rigidly bind to the nanoparticle. And because it does not rigidly bind to the nanoparticle, you can see that it will be engulfed by the membrane. Whereas when pH is equal to pKa, there's very, very strong adsorption of polymer as is seen. You can see these dots versus these dots. So polymer is adsorbed very tightly onto the nanoparticle, so it will not allow it to interact with the membrane. So as a result, they'll bounce back. So what has also been shown, the same time frame from 0.09 microseconds to 18 microseconds for all 3 blocks. And C is the case when pH is high. So when pH is high, meaning pH is higher than pKa, the polymer does get adsorbed, but it does not very strongly get adsorbed. And because pH is high, in fact, you can see that there is interatomic difference between these polymer to polymer bonds. That means there's electrostatic repulsion happening. So because of that, this particular C case acts somewhere between B and A. So engulfing does happen, but definitely it is less than the B part. So then coming to dendrimers and dendrons, which is another delivery system. Dendrimers are simple self-assembled systems. And in fact, this Z0, G1, G2 indicates the generation. These are surfactant molecules which self-assemble. So they'll go on increasing in size this way. But believe me, when you measure, their size is in nanometer range. So for dendrons, you can study interaction with other molecules. You can look at effect of solvents, different counter ions, generation number, etc. And also one could modify the terminal groups. So typically you have terminal groups which are either amino or you will have OH groupings. So you can look at, if I change that terminal group, how will the total structure of dendrimer change and how in fact it will interact with my API in question. And this is one example which is of dendrimer, wherein it is shown that how dendrimer interacts with DNA. So you can see that G3, that is third generation of dendrimer, there is minute deformation in DNA. Whereas in the case of G4, both the DNA as well as the dendrimer are deformed. Whereas in this case, the C case, DNA is deformed, but dendrimer stays put. So very easy terms. In fact, it is shown even schematically here. This is molecular dynamic study, but schematically also it is shown that here the dendrimer remains same, but it changes the DNA. And in this case, there is little change which is happening, in fact, to DNA as well as the dendrimer and so on. So such interactions easily can be checked in silico. Not only that, one can look at mechanism of endosomal escape, which depends on pH responsive dendrimer. So you can see that once a nanoparticle, which is a dendrimer, you know that extracellular fluid has pH of 7, so it will be taken in by endocytosis. So when the pH decreases, that means when the pH becomes 5, which typically is the case, because once a person has, say, cancer, the pH in the cells decreases, which becomes acidic. So at that acidic pH, the dendrimer in fact swells. And this is, you can see here, it's very easily shown at physiological pH. This is how it will interact. At endosomal formation, when the pH starts changing, there's little more interaction which is taking place. And when completely endosome has ruptured, that means the dendrimer actually has reached the nucleus. This will be shown by molecular dynamic study. So you can actually visualize this in silico. So if you can visualize it in silico, obviously it is going to happen in vivo also. So obviously in vivo, one needs to do it using confocal, or you need to look at fluorescent tagging and then track it, what happens within the cells. And now these are some of the examples from our published data, wherein we have looked at, can we improve ocular mucoretention? We all know that eye drops have to be instilled into eye for a number of times to elicit their action. Specifically, if you have, say, glaucoma, or if you have certain infection, wherein physician will tell you that to put every 3 hours. It's so time consuming. So our hypothesis was, can we interact our drug in question, which was norfloxacin, with the polymer and make drug polymer conjugates, and whether those drug polymer conjugates will react with this mucin layer. As you know that if you look at the structure of eye, you do have cornea, followed by mucin layer, then you have aqueous humor, and then the outermost layer is lipidic. So obviously, if you want your dosage form to be retained and yet be effective, it has to cross lipid, but it has to get retained in this mucoaqueous layer. To retain it, our hypothesis was if I make a drug polymer complex, whether it will interact with mucin and whether I can see that interaction with ocular surface protein, mucin in this case, in silico. So we did this study, looked at binding interactions which occur on the ocular surface. And also what we did is we made a drug polymer conjugate, which was, of course, a covalent. So we don't want this complex to break because we want prolonged antibacterial action. So drug would diffuse out of this polymeric, in fact, conjugate over a period of time. So we did a facile synthesis. In fact, it was only a one-step reaction. And this also indicates how interdisciplinary research is good. I'm from pharmaceutics. All this I have learned only by way of interest. And in fact, many a times it is blessing to learn from students who would want to work on such problems. So we get students, in fact, in our lab of pharmaceutics, I have students from varied backgrounds. We have pharmacologists, we have pharmaceutical chemists, and we also have biotechnologists. So it becomes easy to work as a team. So we synthesized a functionalized polymer. So in fact, norfloxacin was functionalized, in fact, was reacted with polymethyl ether malic acid. So it gave a conjugate. And we perform molecular docking studies, and you can see now the molecular docking results. So I have given it by two different methods. One is by quick prop and one by Jaguar. But both are indicating that norfloxacin as a drug, if it has PSA score of about 97, when it forms a complex, it increases to 306. Same is the case with Jaguar also. It is clearly showing that there is the change in property. So if there is a change in property, whether there is interaction taking place and how does it react with mucin. So we looked at how it reacts with mucin, and this particular diagram, which is showing the ligand interaction, very clearly shows that it reacts with mucin at different amino acid residues. And it will very clearly give you an arrow with which particular amino acid it is going to react. So if you just take norfloxacin with mucin, there are hardly one or two arrows which you will see in this full diagram. But if you take norfloxacin, which is conjugated with polymethyl ether malic acid, and then look at its interaction, you will see number of these arrows. This also indicates, meaning visually itself it indicates, without looking at these values, it indicates that interaction with mucin is going to be much more here. And with mucin, it will form a three-dimensional structure. And then once we did this, we looked at whether really it gives us a gel and whether really it gives us release profile which is prolonged. So you can see that what had to be administered every 3 hours now will give us release for over 30 hours, 100% in about say 24 hours. So that means it becomes once daily application. So what we did is we made a conjugate. We did in silico studies. From in silico studies, we established that it interacted well with mucin, which is a protein. So our hypothesis was working correct, that it will give us release for prolonged period. So our second step was, of course, to formulate it. So we formulated it as a aqueous gel, in fact, because it was a polymer. So it gave us a in situ kind of a gel, and put all the other ingredients to stabilize this particular formulation, and then looked even at muco-adhesive strength. So you can see that sodium CMC, which is a known muco-adhesive agent, we have compared it with that. So drug doesn't have muco-adhesion. Our polymer does have muco-adhesion, and that's the reason why we selected that polymer. But when you're taking drug along with the polymer conjugate, it is surpassing even sodium CMC's muco-adhesion. So not only in silico, but by doing in vitro studies, and now in fact in vivo studies are going on. We have finished in vitro antibiotic assays. Now in vivo is going on because we had to look at the toxicity of this particular thing before we could do in vivo. So results are really very encouraging. The next one was Xyloglucan. We have many patents in this direction. In fact, this was the polymer based on which we developed a vaccine, and that got grant from Bill Melinda Gates, and now we have proposed vaccine for COVID, and DBT has approved that project also. So it is a tamarind seed derived xyloglucan. What I've done in this study was, my hypothesis was, can I make using a natural ingredient, which is a waste material? Tamarind seed is a waste material. There's no use. So can I take that and make really good use of it? So can I make hydrogel, which are not only multifunctional, but multi-responsive also? So for that, I had to study which of the co-solvents will give gelling property with this xyloglucan. So obviously in water, we want the system, because again, this system also we were trying, in fact, for ocular use also. Ocular films we were making, but not only ocular use. Now I told you that we have used it even for vaccine purposes. So mechanistic evaluation of gel network we had to see. So we looked at molecular dynamic studies with different types of, in fact, co-solvents, and looked at their trajectories. Trajectory is what path it will follow when it interacts atom to atom. So as you can see that with oleic acid, we were trying to make an emulsion of it. So this has not given us good results. Neither has polysorbate 60 given us results. But this is what, which I have enlarged it here, has given us very good results. So when you are using glycerol as a gelator with a dimeric xyloglucan in presence of water, in 100 picosecond, you can see the strong HOH bonding. And how do you know it is strong? If you have, this particular distance less than 2 Armstrong, this would be shown as a very, very strong bonding. And that is what we wanted. So less than 2 Armstrong. So we got it with glycerol. And therefore, for oleic acid based emulsion, we use glycerol, water, oleic acid, and xyloglucan. So molecular level associations and xyloglucan based hydrogels could control and preserve. Believe me, it was nano emulsion. And you know how difficult it is to stabilize nano emulsion. Nano emulsions are not thermodynamically stable. Nano emulsions are kinetically stabilized emulsions in nanometric range. So this kinetically stabilized meaning either you are giving high shear or you're giving high temperature for their formation. So these need to be stabilized, at least have 2 years stability so that can be used. We could easily, using in silico, find out what ingredients would be best. And this is, I think, the last study wherein we have looked at phytopolyphenols and their GP120 CD4 binding inhibitory properties. Our hypothesis here is, this study, we were, in fact, this was also a DBT ICMR sponsored project wherein we have proposed use of natural materials as anti-HIV as well as microbicides. So what was our hypothesis? Typically, the virus will have, this G120 and receptor has CD4. So you need to find out ligand which binds to the hydrophobic region of GP120. So if it binds to the hydrophobic region of GP120, it will then prevent this binding, CD4. That means we were looking at fusion inhibitors. So which can be applied, say nowadays, vaginal microbicide or vaginal gels, which can be applied by those people who want protection, specifically, sex workers who need protection. And also now that homosexuals are very common, we formulated even a rectal. In fact, the DBT sanctioned the project for rectal microbicide. So the gel could be applied both rectally and vaginally. So hypothesis was, find out what it would be which will actually get into the domains of GP120 in the hydrophobic domain, so that it can prevent CD4 receptor binding. The receptor, of course, we imported from PDB, protein database, and that was 1RZ1. So we did in silico screening and we did in vitro screening. And on this basis, we have got excellent results. In fact, for theaflavin is nothing but, you know, that theaflavin and ECGC are from green tea and black tea. They are the actives coming from these tea. Believe me, they are more efficacious, less toxic, and have lower sensitivity to resistance when compared with the antiviral, which is commonly used, and that is tenofovir. So you can have natural ingredient based gel or any kind of system which can be delivered vaginally or rectally to act as anti-HIV, and you do not have to use a synthetic moiety. So this is going to reduce cost, and therefore it is also going to actually increase patient compliance, because not only they act as antiviral, they have properties as anti-cancer, they can be used as antioxidants. So overall health also is going to improve. So we looked at the MD simulation. So this is how the structure of HIV1 GP120 looks. It's a huge structure. So you'll get confused if you look at different colors. Here, different colors actually represent different type of atoms. But it's very simple to look at this interaction. And therefore, your Schrodinger becomes important because you're looking at the ligand-based interactions. So here you can very clearly see this is a binding pose of curcumin. This is a binding interaction. It's showing curcumin with GP120. This is of tetrahydrocurcumin, which is analog of curcumin, and this is for binding interaction with THC. So very clearly it is shown that THC has higher binding affinity towards, not only it shows different arrows, you know, it also shows to which amino acid it has affinity. And if this amino acid, it has affinity, it forms a complex and goes and sits into pocket of the receptor, obviously it will be effective. And this glide score clearly indicates. Look at the glide score. We need to validate it. So from validating, you have to take known molecule. BMS stands for Bristol-Myers Squibb. So Bristol-Myers Squibb derivative, which was under clinical trial, had a glide score of minus 5.185. Its derivative had glide score of minus 6.1. And now look at our curcumin at THC. THC is even better in glide score than this BMS derivative, which is synthetic. So what we are trying to show you here is that you can, the same way, no, you can scan for even COVID, and that's what our undergraduates have done. And there's one paper on THC being used for even COVID-19. It has given a very good glide score. And if it gives very good glide score, meaning it sits in that pocket very well. And if it sits in that pocket of receptor very well, that means it interacts with that virus very well. And obviously, then it will give its useful property. So you need to select lowest energy conformations. And it has shown that how curcumin interacts with TRP427 and THC interacts both with ASP as well as TRP. So THC was much more useful. So I would like to conclude here. I think by these case studies, you know that the scope of this particular field is huge. And sitting in your four walls at your home, you can do so much using initially the free softwares. And then, of course, the institutes can buy the softwares like Schrodinger to help you do much more. So analogous to the paradigm shift of drug development in the past 3 decades by computer-aided drug design, computational pharmaceutics also has great potential. So that will shift the paradigm of drug delivery research. So now no one is going to go directly, you want to do micelles, you are going, then trying different surfactants. You want to make microemulsion, you're trying different oils, surfactants, co-surfactant. All that can be actually done in silico in matter of nanoseconds. And then you can look at what actually the screened ones to take to the lab. So these are our research areas. And in fact, all research areas, I have some studies, but for lack of time, I have given you some representative studies. And we are working in almost all the area, majorly focusing now on lipid nanocarrier based DDS, because they are having good potential both in prophylaxis as well as actually in treatment. These are our technology platforms. All of them have been patented. Many of them have been actually given to industry, and students, we are lucky that we get very, very good students, and these are some of the technologies transferred. And as Professor Madan had indicated, these stents, which are devices, drug-coated devices, they are being sold in more than 60 countries. In fact, we have many US, European PCT patents, and these represent some of our potential collaborators. And without acknowledging them, I cannot move ahead, because they have really supported us in a big way so that we can do good research. These are two of our books which have focused on nanoparticulate drug delivery and pharmaceutical product development. This has been indicated even in the PCI syllabus, because this goes the industry way, in fact, how the product is developed, how the QBD is taken into consideration, everything has been covered in the book. And this is our small lab. Mumbai space is a problem, but I have very, very good students, and all the facilities, whatever you would like to name, have been generated through government grants and industrial grants. And we work with single-mindedness. And our motto is, take up one idea, make that idea your life, think of it, dream of it, live on that idea. Let the brain, muscles, nerves, every part of your body be full of that idea. Just leave any other idea alone. That's the way to success. In a nutshell, what Swami Vivekananda is trying to tell us is focus. So once we select a particular problem, we see that we reach the end. Should not change the problem, you can change the ways to reach problem. And this is my nano group, and I would fail in my duty if I do not acknowledge Anjali Pandya and Shrikant, who have helped in putting the matter for this particular presentation together. And I would end by saying thank you. Secret to getting ahead is getting started. So in case, even if 1% of people I have motivated, I would feel that they should take up this area, which is very, very convenient and easy way to go a long way. Thank you so much. And I'm open to any questions which you might have. You are on mute, Professor Madan.
Any question from our participant side? I think no questions. Okay, thank you very much, ma'am. This is very nice information provide to our participant, and maybe this type of information will be very, very useful for researcher, and in a very effective way you send this information, so maybe these researchers will be useful in future. So on the behalf of Shobit University, we are very thankful to you to provide to us your valuable time, and all the panel members and all the participants. Thank you very much.
It is my pleasure to be of some use. So I hope that people will at least download the free softwares and start using it.
Yes, even though they can contact on you, whatever is they
They
require.
can contact me anytime. In fact, by way of mail or even my phone number, you can share with them. Anyone has any question, anytime they can contact.
Thank you.
Thank
very
you,
much.
ma'am.
Thank you, sir. It was
We'll be in
pleasure.
touch. Thank you very much.
Okay, bye bye.